Welcome to today's Explainer, where we are going to explore the high-tech, completely invisible communication network that is literally keeping you alive right now. We're talking about signal transduction pathways. It's basically this fascinating, high-speed biological internet happening inside your own body at this very second.
Okay, let's just dive right into this by asking a massive, mind-boggling question. How do the trillions of individual cells in your body actually manage to perfectly coordinate everything? From your heartbeat to your immune system, how do they know what to do? I mean, think about it. Your body isn't just a single solid block of clay, right? It is a massive, bustling metropolis of microscopic units. And without a way to talk to each other, it would just be pure chaos. They desperately need a system to detect the environment around them and then convert that information into a highly specific biological response.
To make sense of all this, we're going to break this complex topic down into 6 distinct pieces: how cells communicate, the 3 steps of signaling, major channels, amplifying the message when signaling fails, and finally, the future of medical tech. So the absolutely crucial point here is understanding this term, signal transduction. What does that actually mean? Well, it's basically how your cells receive a message from the outside world and translate it into real biological action on the inside. You can kind of think of it like receiving a text message on your phone that tells you to start running. Your eyes read the signal, sure, but your legs actually do the physical work. The cell pulls this off through an incredible, complex cascade of molecular events.
Section 1. How cells communicate and the components of the network.
So if you want to build a communication network, you're going to need 4 key parts. Think of the first one, the signaling molecule, also known as the ligand, like a hormone or a growth factor, as the incoming text message itself. Number 2 is the receptor. This is a highly specialized protein sitting on the cell membrane or sometimes hidden inside the cell, and it acts exactly like a cell tower, just waiting to catch that specific message. Third, we have the signal relay molecules, like protein kinases. These are basically the fiber optic cables that transmit the data deep inside the cell. And finally, number 4, the effector molecules. These are things like transcription factors or metabolic enzymes that actually produce the final cellular response. It's the action your phone takes when the message is fully processed and understood.
Section 2. The 3 steps of signaling, or what I like to call the cellular game of telephone.
This whole process always follows 3 super elegant steps. Step 1 is reception. This is the moment our ligand actually docks and perfectly binds to its specific receptor. Step 2 is transduction. Just like a giant game of telephone, the receptor changes shape and passes the signal along a cascade of molecular events. One thing triggers the next, which triggers the next. Finally, step 3 is the response, where the cell actually executes a specific action, like producing a new protein or starting cell division. To put that into perspective with a real-world example, when you eat a big meal, the insulin molecule arrives at reception. The message is passed along via transduction, and the final response, which is your cells absorbing glucose for energy, is perfectly executed.
Moving on to Section 3, major cellular communication channels and the specific pathways. First up is the GPCR pathway, or G-protein coupled receptor. This is one of the absolute largest receptor families in your cells. You can think of it as the main broadband network for hormones, neurotransmitters, and sensory molecules. When a ligand binds here, a G-protein activates and pumps out what we call second messengers. These are tiny molecules like CAMP, which activates protein kinase A, or calcium ions that actually trigger your muscles to contract. These second messengers really do the heavy lifting to get the cellular job done incredibly fast.
And that leads us to our next major highway, the RTK pathway, or receptor tyrosine kinase. Now, while GPCR handles a lot of sensory and hormone data, RTK regulates the big stuff. We're talking cell growth, differentiation, and survival. When a growth factor binds here, the receptors literally pair up. They form what's called a dimer and become phosphorylated. This creates a massive master control switch. A huge example of this is the MAPK pathway, specifically the Ras-Raf-Mec-ERK sequence, which is a primary driver for cell proliferation and overall development.
But wait, there are other highly specialized channels you really should know about. We've got the JK-STAT pathway, which is absolutely critical for your immune regulation and blood cell formation. There's the Wnt pathway, which controls embryonic development and keeps your stem cells maintained. The Notch pathway is essential for nervous system development and literally deciding a cell's ultimate fate. And finally, TGF-beta, which is in charge of cell growth control and tissue repair. Each of these is a dedicated, specific biological frequency, ensuring your body responds perfectly to whatever environment it's in.
Section 4. Amplifying the message, from 1 to thousands. So you might be wondering, How do these tiny microscopic messages not just get lost in the noise? The answer is signal amplification. A single, solitary external signal can produce a massive internal response. Just one hormone molecule binding to one single receptor can activate multiple enzymes, and then those enzymes can go on to activate thousands of downstream molecules. It creates a rapid, exponential biological boom. This is amazing because it allows your cells to respond incredibly efficiently without needing millions of hormone molecules flooding your system just to get a basic reaction.
So how is this rapid amplification actually controlled, without spiraling out of control? It all comes down to two vital enzymes working as microscopic on and off switches. On one hand, we have protein kinases. These enzymes grab ATP and slap a phosphate group right onto a protein. This phosphorylation activates the protein or totally changes its function. It is the ultimate on switch. But, you can't leave the switch on forever, right? That's where protein phosphatases come in on the other side. They step in and strip those phosphate groups away, effectively turning off the signaling pathway and restoring normal baseline cellular conditions. It's just a beautifully balanced system of activation and deactivation.
Section 5. When cell signaling fails, or what happens when the network goes down.
Unfortunately, things do break, and many cancers are fundamentally the result of abnormal cell signaling. Remember the RTK pathways we mentioned a minute ago that control growth? Well, if you have an overactivation of EGFR, that's the epidermal growth factor receptor, it can cause abnormal, out-of-control increases in cell division and tumor growth. Similarly, mutations in the RAS protein can cause it to get basically jammed in the on position. That sends a continuous, non-stop growth signal to the cell, which is a massive primary driver of cancer.
But it isn't just cancer. Defective signaling is literally the root cause of diabetes, where the insulin message is basically dropped by the network, leading to reduced glucose uptake. Over in your immune system, abnormal cytokine signaling can result in a massive network misfire that causes chronic inflammation and autoimmune diseases. And in your brain, changes in these pathways can impair neuron survival, leading to really debilitating neurological diseases. When this cellular communication breaks down, the whole metropolis falls into disarray.
Section 6. The Future of Medical Tech. We are hacking the network.
Because we now understand these pathways, scientists are quite literally hacking this communication network. In drug discovery, they're developing highly targeted kinase inhibitors that specifically hunt down and block those broken on switches in cancer. In cancer therapy, we have treatments like EGFR and HER2 inhibitors that shut down abnormal signaling pathways. In regenerative medicine, researchers are learning how to control stem cell differentiation to actually regenerate damaged tissue. And in synthetic biology, which is wild, scientists are engineering completely artificial signaling systems to program cells for brand new biotechnology applications. We aren't just passively observing the network anymore, we are actively reprogramming it.
And honestly, the future is even wilder. Researchers are now utilizing artificial intelligence to map out and predict these incredibly complex, multi-layered signaling networks. By leveraging network biology and bioinformatics, we are stepping into an era of entirely personalized cancer therapy and true precision medicine. Imagine a world where a treatment is designed precisely for how your specific, unique cellular network is communicating.
Let's wrap things up with the conclusion and a quick revision, tying it all together.
Let's just quickly review your core components, the true hardware of the system. Remember this cheat sheet. The ligand sends the message, the receptor catches it at the membrane. The kinases acts as the on switch by slapping on phosphates, while phosphatases turn it off. Second messengers transmit that signal rapidly inside the cell, and finally, transcription factors execute the ultimate biological order by regulating your gene expression.
And these are the major cellular highways we travel today. From the massive GPCR pathway handling your hormones and sensory data to the RTK and MACPAC sequences driving cell division. We've got JSTAT running your immune defense, WNT maintaining your stem cells, Notch deciding cell fate, and TGF-beta controlling tissue growth and repair. It is an absolute masterpiece of biological engineering.
By understanding these microscopic signal transduction pathways, we are no longer just putting band-aids on surface symptoms. We are intervening at the very root of cellular communication. We are quite literally rewriting the software of life itself, which leaves us with one incredible, awe-inspiring question. If we can engineer artificial cellular signals from scratch, what completely incurable diseases will we rewrite the code for next? Thanks so much for joining me on this explainer, and keep your curiosity switched on.